Section 22 of Gray's Anatomy, Part 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Mark Roche. Anatomy of the Human Body, Part 5, by Henry Gray. Chapter 2H. The Large Intestine. Intestinum crassum. The sigmoid colon sigmoideum, pelvic colon, sigmoid flexure, forms a loop which averages about 40 centimeters in length and normally lies within the pelvis, but on account of its freedom of movement is liable to be displaced into the abdominal cavity. It begins at the superior aperture of the lesser pelvis, where it is continuous with the iliac colon, and passes transversely across the front of the sacrum to the right side of the pelvis. It then curves on itself and turns toward the left to reach the middle line at the level of the third piece of the sacrum, where it bends downward and ends in the rectum. It is completely surrounded by peritoneum, which forms a mesentery, sigmoid mesocolon, which diminishes in length from the center toward the ends of the loop, where it disappears so that the loop is fixed at its junctions with the iliac colon and rectum, but enjoys a considerable range of movement in its central portion. Behind the sigmoid colon are the external iliac vessels, the left piriformis and left sacral plexus of nerves. In front, it is separated from the bladder in the male and the uterus in the female by some coils of the small intestine. The rectum, intestinum rectum, is continuous above with the sigmoid colon, while below it ends in the anal canal. From its origin at the level of the third sacral vertebra, it passes downward, lying in the sacrococcygeal curve, and extends for about 2.5 centimeters in front of and a little below the tip of the coccyx, as far as the apex of the prostate. It then bends sharply backward into the anal canal. It therefore presents two antero-posterior curves, an upper with its convexity backwards, and the lower with its convexity forward. Two lateral curves are also described, one to the right opposite the junction of the third and fourth sacral vertebra, and the other to the left, opposite the left sacrococcygeal articulation. They are, however, of little importance. The rectum is about 12 centimeters long, and at its commencement its caliber is similar to that of the sigmoid colon, but near its termination it is dilated to form the rectal ampulla. The rectum has no saculations comparable to those of the colon, but when the lower part of the rectum is contracted, its mucous membrane is thrown into a number of folds, which are longitudinal in direction, and are effaced by the distension of the gut. Besides these, there are certain permanent transverse folds of a semilunar shape, known as Houston's valves. They are usually 3 in number, sometimes a fourth is found, and occasionally only 2 are present. One is situated near the commencement of the rectum, on the right side. A second extends inward from the left side of the tube, opposite the middle of the sacrum. A third, the largest and most constant, projects backward from the forepart of the rectum, opposite the fundus of the urinary bladder. When a fourth is present, it is situated nearly 2.5 centimeters above the anus on the left and posterior wall of the tube. These folds are about 12 millimeters in width, and contain some of the circular fibers of the gut. In the empty state of the intestine they overlap each other, as Houston remarks, so as effectually as to require considerable maneuvering to conduct a bougie or the finger along the canal. Their use seems to be to support the weight of fecal matter and prevent its urging toward the anus, where its presence always excites a sensation of demanding its discharge. Footnote. Patterson, The Form of the Rectum, Journal of Anatomy and Physiology, Volume 43, utilizes the third fold for the purpose of dividing the rectum into an upper and lower portion. He considers the latter to be just as much a duct as the narrower anal canal below, and maintains that under normal conditions it does not contain feces except during the act of defecation. End footnote. The peritoneum is related to the upper 2/3 of the rectum, covering at first its front and sides, but lower down its front only. From the latter it is reflected onto the seminal vesicles in the male and the posterior vaginal wall in the female. The level at which the peritoneum leaves the anterior wall of the rectum to be reflected onto the viscus in front of it is of considerable importance from a surgical point of view, in connection with the removal of the lower part of the rectum. It is higher in the male than in the female. In the former, the height of the rectovesical excavation is about 7.5 centimeters, i.e., the height to which an ordinary index finger can reach from the anus. In the female, the height of the rectouterine excavation is about 5.5 centimeters from the anal orifice. The rectum is surrounded by a dense tube of fascia derived from the fascia endopelvina, but fused behind with the fascia covering the sacrum and coccyx. The fascial tube is loosely attached to the rectal wall by areolar tissue in order to allow the distension of the viscus. Relations of the rectum. The upper part of the rectum is in relation behind with the superior hemorrhoidal vessels, the left piriformis, and left sacral plexus of nerves, which separate it from the pelvic surfaces of the sacral vertebra. In its lower part, it lies directly on the sacrum, coccyx, and levatoris ani, a dense fascia alone intervening. In front, it is separated above, in the male, from the fundus of the bladder, in the female from the intestinal surface of the uterus and its appendages by some convolutions of the small intestine, and frequently by the sigmoid colon. Below, it is in relation in the male with the triangular portion of the fundus of the bladder, the vesiculae seminales and ductus deferentes, and more anteriorly with the posterior surface of the prostate. In the female, with the posterior wall of the vagina. The anal canal, pars analis recti, or terminal portion of the large intestine, begins at the level of the apex of the prostate, is directed downward and backward, and ends at the anus. It forms an angle with the lower part of the rectum, and measures from 2.5 to 4 centimeters in length. It has no peritoneal covering, but is invested by the sphincter ani internus, supported by the levatoris ani, and surrounded at its termination by the sphincter ani externus. In the empty condition it presents the appearance of an anterio-posterior longitudinal slit. Behind it is a mass of muscular and fibrous tissue, the anococcygeal body, Symington. In front of it, in the male, but separated by connective tissue from it, are the membranous portion and bulb of the urethra, and the fascia of the urogenital diaphragm. And in the female, it is separated from the lower end of the vagina by a mass of muscular and fibrous tissue, named the perineal body. The lumen of the anal canal presents, in its upper half, a number of vertical folds, produced by an infolding of the mucous membrane and some of the muscular tissue. They are known as the rectal columns, Morgagni, and are separated from one another by furrows, rectal sinuses, which end below in small valve-like folds termed anal valves, which join together the lower ends of the rectal columns. Structure of the colon. The large intestine has 4 coats: serous, muscular, areolar, and mucous. The serous coat, tunica serosa, is derived from the peritoneum, and invests the different portions of the large intestine to a variable extent. The cecum is completely covered by the serous membrane, except in about 5% of cases where the upper part of the posterior surface is uncovered. The ascending, descending, and iliac parts of the colon are usually covered only in front and at the sides. A variable amount of the posterior surface is uncovered. The transverse colon is almost completely invested, the parts corresponding to the attachment of the greater omentum and transverse mesocolon being alone excepted. The sigmoid colon is entirely surrounded. The rectum is covered above on its anterior surface and sides, below on its anterior aspect only. The anal canal is entirely devoid of any serous covering. In the course of the colon, the peritoneal coat is thrown into a number of small pouches filled with fat, called appendices epiploici. They are most numerous on the transverse colon. The muscular coat, tunica muscularis, consists of an external longitudinal and an internal circular layer of non-striped muscular fibers. The longitudinal fibers do not form a continuous layer over the whole surface of the large intestine. In the cecum and colon they are especially collected into 3 flat longitudinal bands, teni coli, each of about 12 mm in width. One, the posterior, is placed along the attached border of the intestine. The anterior, the largest, corresponds along the arch of the colon to the attachment of the greater omentum, but is in front in the ascending, descending, and iliac parts of the colon, and in the sigmoid colon. The third, or lateral band, is found on the medial side of the ascending and descending parts of the colon, and on the under aspect of the transverse colon. These bands are shorter than the other coats of the intestine, and serve to produce the saculi, which are characteristic of the cecum and colon. Accordingly, when they are dissected off, the tube can be lengthened and its saculated character disappears. In the sigmoid colon, the longitudinal fibers become more scattered, and around the rectum they spread out and form a layer, which completely encircles this portion of the gut, but is thicker on the anterior and posterior surfaces, where it forms 2 bands, than on the lateral surfaces. In addition, 2 bands of plain muscular tissue arise from the second and third coccygeal vertebrae, and pass downward and forward to blend with the longitudinal muscular fibers on the posterior wall of the anal canal. These are known as the rectococcygeal muscles. These circular fibers form a thin layer over the cecum and colon, being especially accumulated in the intervals between the saculi. In the rectum they form a thick layer, and in the anal canal they become numerous and constitute the sphincter ani internus. The areolar coat, tela submucosa, submucous coat, connects the muscular and mucous layers closely together. The mucous membrane, tunica mucosa, in the cecum and colon is pale, smooth, destitute of villi, and raised into numerous crescentic folds, which correspond to the intervals between the saculi. In the rectum it is thicker, of a darker color, more vascular, and connected loosely to the muscular coat, as in the esophagus. As in the small intestine, the mucous membrane consists of a muscular layer, the muscularis mucosae, a quantity of retiform tissue in which the vessels ramify, a basement membrane and epithelium which is of the columnar variety, and resembles the epithelium found in the small intestine. The mucous membrane of the large intestine presents for examination glands and solitary lymphatic nodules. The glands of the great intestine are minute tubular prolongations of the mucous membrane arranged perpendicularly side by side over its entire surface. They are longer, more numerous, and placed in much closer apposition than those of the small intestine, and they open by minute rounded orifices upon the surface, giving it a cribriform appearance. Each gland is lined by short columnar epithelium and contains numerous goblet cells. The solitary lymphatic nodules, noduli lymphatic solitarii, of the large intestine are most abundant in the cecum and vermiform process, but are irregularly scattered also over the rest of the intestine. They are similar to those of the small intestine. Vessels and nerves. The arteries supplying the colon are derived from the colic and sigmoid branches of the mesenteric arteries. They give off large branches, which ramify between and supply the muscular coats, and after dividing into small vessels in the submucous tissue, pass to the mucous membrane. The rectum is supplied by the superior hemorrhoidal branch of the inferior mesenteric, and the anal canal by the middle hemorrhoidal from the hypogastric, and the inferior hemorrhoidal from the internal pudendal artery. The superior hemorrhoidal, the continuation of the inferior mesenteric, divides into 2 branches, which run down either side of the rectum to within about 12.5 centimeters of the anus. They here split up into about 6 branches, which pierce the muscular coat and descend between it and the mucous membrane in a longitudinal direction, parallel with each other as far as the sphincter ani internus, where they anastomose with the other hemorrhoidal arteries and form a series of loops around the anus. The veins of the rectum commence in a plexus of vessels which surrounds the anal canal. In the vessels forming this plexus are smaller saccular dilations, just within the margin of the anus. From the plexus, about 6 vessels of considerable size are given off. These ascend between the muscular and mucous coats for about 12.5 centimeters, running parallel to each other. They then pierce the muscular coat, and, by their union, form a single trunk, the superior hemorrhoidal vein. This arrangement is termed the hemorrhoidal plexus. It communicates with the tributaries of the middle and inferior hemorrhoidal veins at its commencement, and thus a communication is established between the systemic and portal circulations. The lymphatics of the large intestine are described on page 711. The nerves are derived from the sympathetic plexuses around the branches of the superior and inferior mesenteric arteries. They are distributed in a similar way to those found in the small intestine. Congenital hernia. There are some varieties of oblique inguinal hernia depending upon congenital defects in the saccus vaginalis, the pouch of peritoneum which precedes the descent of the testes. Normally, this pouch is closed before birth, closure commencing at 2 points, viz., at the abdominal inguinal ring and at the top of the epididymis, and gradually extending until the whole of the intervening portion is converted into a fibrous cord. From failure in the completion of this process, variations in the relation of the hernial protrusion to the testes and tunica vaginalis are produced. These constitute distinct varieties of inguinal hernia, viz., the hernia of the funicular process and the complete congenital variety. Where the saccus vaginalis remains patent throughout, the cavity of the tunica vaginalis communicates directly with that of the peritoneum. The intestine descends along this pouch into the cavity of the tunica vaginalis, which constitutes the sac of the hernia, and the gut lies in contact with the testes. Though this form of hernia is termed complete congenital, the term does not imply that the hernia existed at birth, but merely that a condition is present which may allow the descent of the hernia at any moment. As a matter of fact, congenital hernia frequently do not appear until adult life. Where the processus vaginalis is occluded at the lower point only, i.e., just above the testes, the intestine descends into the pouch of the peritoneum as far as the testes, but is prevented from entering the sac of the tunica vaginalis by the septum which has formed between it and the pouch. This is known as hernia into the funicular process, or incomplete congenital hernia. It differs from the former in that instead of enveloping the testes, it lies above it. End of section 22. Recording by Mark Roche, Tokyo, Japan.
Of Anatomy of the Human Body, Part 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body, Part 5, by Henry Gray. The Liver, Part 1. Hepar. The liver, the largest gland in the body, has both external and internal secretions, which are formed in the hepatic cells. Its external secretion, the bile, is collected after passing through the bile capillaries by the bile ducts, which join like the twigs and branches of a tree to form 2 large ducts, which unite to form the hepatic duct. The bile is either carried to the gallbladder by the cystic duct, or poured directly into the duodenum by the common bile duct where it aids in digestion. The internal secretions are concerned with the metabolism of both nitrogenous and carbohydrate materials absorbed from the intestine and carried to the liver by the portal vein. The carbohydrates are stored in the hepatic cells in the form of glycogen, which is secreted in the form of sugar directly into the bloodstream. Some of the cells lining the blood capillaries of the liver are concerned in the destruction of red blood corpuscles. It is situated in the upper and right parts of the abdominal cavity, occupying almost the whole of the right hypochondrium, the greater part of the epigastrium, and not uncommonly extending into the left hypochondrium as far as the mammillary line. In the male it weighs from 1.4 to 1.6 kilograms, in the female from 1.2 to 1.4 kilograms. It is relatively much larger in the fetus than in the adult, constituting in the former about 1/18th and in the latter about 1/36 of the entire body weight. Its greatest transverse measurement is from 20 to 22.5 centimeters. Vertically, near its lateral or right surface, it measures about 15 to 17.5 centimeters, while its greatest antero-posterior diameter is on a level with the upper end of the right kidney and is from 10 to 12.5 centimeters. Opposite the vertebral column, its measurement from before backward is reduced to about 7.5 centimeters. Its consistence is that of a soft solid. It is friable, easily lacerated, and highly vascular. Its color is a dark reddish brown, and its specific gravity is 1.05. To obtain a correct idea of its shape, it must be hardened in situ, and it will then be seen to present the appearance of a wedge, the base of which is directed to the right, and the thin edge toward the left. Symington describes its shape as that of a right-angled triangular prism with the right angle rounded off. Surfaces. The liver possesses three surfaces, namely superior, inferior, and posterior. A sharp, well-defined margin divides the inferior from the superior in front. The other margins are rounded. The superior surface is attached to the diaphragm and anterior abdominal wall by a triangular falciform fold of peritoneum, the falciform ligament, in the free margin of which is a rounded cord, the ligamentum teres, obliterated umbilical vein. The line of attachment of the falciform ligament divides the liver into two parts, termed the right and left lobes, the right being much the larger. The inferior and posterior surfaces are divided into four lobes by 5 fossae, which are arranged in the form of the letter H. The left limb of the H marks on these surfaces the division of the liver into right and left lobes. It is known as the left sagittal fossa, and consists of two parts, namely, the fossa for the umbilical vein in front and the fossa for the ductus venosus behind. The right limb of the H is formed in front by the fossa for the gallbladder and behind by the fossa for the inferior vena cava. These two fossae are separated from one another by a band of liver substance termed the Caudate Process. The bar connecting the two limbs of the H is the porta, transverse fissure. In front of it is the quadrate lobe, behind it the caudate lobe. The superior surface, facies superior, comprises a part of both lobes, and as a whole is convex, and fits under the vault of the diaphragm, which in front separates it on the right from the 6th to the 10th ribs and their cartilages, and on the left from the 7th and 8th costal cartilages. Its middle part lies behind the xiphoid process, and in the angle between the diverging rib cartilage of opposite sides is in contact with the abdominal wall. Behind this the diaphragm separates the liver from the lower part of the lungs and pleurae, the heart and pericardium, and the right costal arches from the 7th to the 11th inclusive. It is completely covered by peritoneum except along the line of attachment of the falciform ligament. The inferior surface, facies inferior, visceral surface, is uneven, concave, directed downward, backward, and to the left, and is in relation with the stomach and duodenum, the right colic flexure, and the right kidney and suprarenal gland. The surface is almost completely invested by peritoneum. The only parts devoid of this covering are where the gallbladder is attached to the liver, and at the porta hepatis, where the two layers of the lesser omentum are separated from each other by the blood vessels and ducts of the liver. The inferior surface of the left lobe presents behind and to the left the gastric impression, molded over the antero-superior surface of the stomach, and to the right of this a rounded eminence, the tuber omentale, which fits into the concavity of the lesser curvature of the stomach and lies in front of the anterior layer of the lesser omentum. The under surface of the right lobe is divided into two unequal portions by the fossa for the gallbladder. The portion to the left, the smaller of the two, is the quadrate lobe, and is in relation with the pyloric end of the stomach, the superior portion of the duodenum, and the transverse colon. The portion of the under surface of the right lobe to the right of the fossa for the gallbladder presents two impressions, one situated behind the other and separated by a ridge. The anterior of these two impressions, the colic impression, is shallow and is produced by the right colic flexure. The posterior, the renal impression, is deeper and is occupied by the upper part of the right kidney and the lower part of the right suprarenal gland. Medial to the renal impression is a third and slightly marked impression, lying between it and the neck of the gallbladder. This is caused by the descending portion of the duodenum and is known as the duodenal impression. Just in front of the inferior vena cava is a narrow strip of liver tissue, the caudate process, which connects the right inferior angle of the caudate lobe to the undersurface of the right lobe. It forms the upper boundary of the epiploic foramen of the peritoneum. The posterior surface, facies posterior, is rounded and broad behind the right lobe, but narrow on the left. Over a large part of its extent it is not covered by peritoneum. This uncovered portion is about 7.5 centimeters broad at its widest part and is in direct contact with the diaphragm. It is marked off from the upper surface by the line of reflection of the upper layer of the coronary ligament, and from the undersurface by the line of reflection of the lower layer of the coronary ligament. The central part of the posterior surface presents a deep concavity which is molded on the vertebral column and crura of the diaphragm. To the right of this, the inferior vena cava is lodged in its fossa between the uncovered area and the caudate lobe. Close to the right of this fossa and immediately above the renal impression is a small triangular depressed area, the suprarenal impression, the greater part of which is devoid of peritoneum. It lodges the right suprarenal gland. To the left of the inferior vena cava is the caudate lobe, which lies between the fossa for the vena cava and the fossa for the ductus venosus. Its lower end projects and forms part of the posterior border of the porta. On the right it is connected with the undersurface of the right lobe of the liver by the caudate process, and on the left it presents an elevation, the papillary process. Its posterior surface rests upon the diaphragm, being separated from it merely by the upper part of the omental bursa. To the left of the fossa for the ductus venosus is a groove in which lies the antrum cardiacum of the esophagus. The anterior border, margo anterior, is thin and sharp, and marked opposite the attachment of the falciform ligament by a deep notch, the umbilical notch, and opposite the cartilage of the ninth rib by a second notch for the fundus of the gallbladder. In adult males, this border generally corresponds with the lower margin of the thorax in the right mammillary line, but in women and children it usually projects below the ribs. The left extremity of the liver is thin and flattened from above downward. Fossae. The left sagittal fossa, fossa sagittalis sinistra, longitudinal fissure, is a deep groove which extends from the notch on the anterior margin of the liver to the upper border of the posterior surface of the organ. It separates the right and left lobes. The porta joins it at right angles and divides it into two parts. The anterior part, or fossa for the umbilical vein, lodges the umbilical vein in the fetus, and its remains, the ligamentum teres, in the adult. It lies between the quadrate lobe and the left lobe of the liver, and is partially bridged over by a prolongation of the hepatic substance, the Pons Hepatis. The posterior part, or fossa for the ductus venosus, lies between the left lobe and the caudate lobe. It lodges in the fetus, the ductus venosus, and in the adult a slender fibrous cord, the ligamentum venosum, the obliterated remains of that vessel. The porta or transverse fissure, porta hepatis, is a short but deep fissure about 5 centimeters long, extending transversely across the undersurface of the left portion of the right lobe, nearer its posterior surface than its anterior border. It joins nearly at right angles with the left sagittal fossa and separates the quadrate lobe in front from the caudate lobe and process behind. It transmits the portal vein, the hepatic artery and nerves, and the hepatic duct and lymphatics. The hepatic duct lies in front and to the right, the hepatic artery to the left, and the portal vein behind and between the duct and artery. The fossa for the gallbladder, fossa vesicae fellei, is a shallow oblong fossa placed on the undersurface of the right lobe, parallel with the left sagittal fossa. It extends from the anterior free margin of the liver, which is notched by it, to the right extremity of the porta. The fossa for the inferior vena cava, fossa venae cavae, is a short, deep depression, occasionally a complete canal in consequence of the substance of the liver surrounding the vena cava. It extends obliquely upward on the posterior surface between the caudate lobe and the bare area of the liver, and is separated from the porta by the caudate process. On slitting open the inferior vena cava, The orifices of the hepatic veins will be seen opening into this vessel at its upper part, after perforating the floor of this fossa. Lobes. The right lobe, Lobus hepatis dexter, is much larger than the left, the proportion between them being as 6:1. It occupies the right hypochondrium and is separated from the left lobe on its upper surface by the falciform ligament, on its under and posterior surfaces by the left sagittal fossa, and in front by the umbilical notch. It is of a somewhat quadrilateral form, its under and posterior surfaces being marked by 3 fossae, the porta and the fossae for the gall bladder and inferior vena cava, which separate its left part into 2 smaller lobes, the quadrate and caudate lobes. The impressions on the right lobe have already been described. The quadrate lobe, lobus quadratus, is situated on the under surface of the right lobe, bounded in front by the anterior margin of the liver, behind by the porta, on the right by the fossa for the gall bladder, and on the left by the fossa for the umbilical vein. It is oblong in shape, its antero-posterior diameter being greater than its transverse. The caudate lobe, lobus caudatus, Spigelian lobe, is situated upon the posterior surface of the right lobe of the liver, opposite the 10th and 11th thoracic vertebrae. It is bounded below by the porta, on the right by the fossa for the inferior vena cava, and on the left by the fossa for the ductus venosus. It looks backward, being nearly vertical in position. It is longer from above downward than from side to side, and is somewhat concave in the transverse direction. The caudate process is a small elevation of the hepatic substance extending obliquely lateralward from the lower extremity of the caudate lobe to the under surface of the right lobe. It is situated behind the porta and separates the fossa for the gall bladder from the commencement of the fossa for the inferior vena cava. The left lobe, lobus hepatis sinister, is smaller and more flattened than the right. It is situated in the epigastric and left hypochondriac regions. Its upper surface is slightly convex and is molded onto the diaphragm. Its undersurface presents the gastric impression and omental tuberosity already referred to. Ligaments. The liver is connected to the undersurface of the diaphragm and to the anterior wall of the abdomen by 5 ligaments. 4 of these, the falciform, the coronary, and the 2 lateral, are peritoneal folds. The 5th, the round ligament, is a fibrous cord, the obliterated umbilical vein. The liver is also attached to the lesser curvature of the stomach by the hepatogastric, and to the duodenum by the hepatoduodenal ligament. The falciform ligament, Ligamentum falciformi hepatis, is a broad and thin antero-posterior peritoneal fold, falciform in shape, its base being directed downward and backward, its apex upward and backward. It is situated in an antero-posterior plane, but lies obliquely so that one surface faces forward and is in contact with the peritoneum behind the right rectus and the diaphragm, while the other is directed backward and is in contact with the left lobe of the liver. It is attached by its left margin to the under surface of the diaphragm and the posterior surface of the sheath of the right rectus, as low down as the umbilicus. By its right margin it extends from the notch on the anterior margin of the liver, as far back as the posterior surface. It is composed of 2 layers of peritoneum, closely united together. Its base or free edge contains between its layers the round ligament and the parumbilical veins. The coronary ligament, Ligamentum coronarium hepatis, consists of an upper and a lower layer. The upper layer is formed by the reflection of the peritoneum from the upper margin of the bare area of the liver to the under surface of the diaphragm, and is continuous with the right layer of the falciform ligament. The lower layer is reflected from the lower margin of the bare area onto the right kidney and suprarenal gland, and is termed the hepatorenal ligament. The triangular ligaments, lateral ligaments, are 2 in number, right and left. The right triangular ligament, Ligamentum triangulare dextrum, is situated at the right extremity of the bare area, and is a small fold which passes to the diaphragm, being formed by the apposition of the upper and lower layers of the coronary ligament. The left triangular ligament, Ligamentum triangulare sinistrum, is a fold of some considerable size, which connects the posterior part of the upper surface of the left lobe to the diaphragm. Its anterior layer is continuous with the left layer of the falciform ligament. The round ligament, Ligamentum teres hepatis, is a fibrous cord resulting from the obliteration of the umbilical vein. It ascends from the umbilicus in the free margin of the falciform ligament to the umbilical notch of the liver, from which it may be traced in its proper fossa on the inferior surface of the liver to the porta, where it becomes continuous with the ligamentum venosum. Fixation of the liver. Several factors contribute to maintain the liver in place. The attachments of the liver to the diaphragm by the coronary and triangular ligaments and the intervening connective tissue of the uncovered area, together with the intimate connection of the inferior vena cava by the connective tissue and hepatic veins, would hold up the posterior part of the liver. Some support is derived from the pressure of the abdominal viscera, which completely fill the abdomen, whose muscular walls are always in a state of tonic contraction. The superior surface of the liver is perfectly fitted to the undersurface of the diaphragm, so that atmospheric pressure alone would be enough to hold it against the diaphragm. The latter, in turn, is held up by the negative pressure in the thorax. The lax falciform ligament certainly gives no support, though it probably limits lateral displacement. End of section 23. Section 24 of Anatomy of the Human Body, Part 5. This LibriVox recording is in the public domain. Anatomy of the Human Body, Part 5, by Henry Gray. The Liver, Part 2. Development. The liver arises in the form of a diverticulum, or hollow outgrowth from the ventral surface of that portion of the gut which afterward becomes the descending part of the duodenum. This diverticulum is lined by entoderm, and grows upward and forward into the septum transversum, a mass of mesoderm between the vitelline duct and the pericardial cavity, and there gives off two solid buds of cells which represent the right and left lobes of the liver. The solid buds of cells grow into columns or cylinders, termed the hepatic cylinders, which branch and anastomize to form a close meshwork. This network invades the vitelline and umbilical veins and breaks up these vessels into a series of capillary-like vessels termed sinusoids, Minot, which ramify in the meshes of the cellular network and ultimately form the venous capillaries of the liver. By the continued growth and ramification of the hepatic cylinders, the mass of the liver is gradually formed. The original diverticulum from the duodenum forms the common bile duct, and from this the cystic duct and the gallbladder arise as a solid outgrowth, which later acquires a lumen. The opening of the common duct is at first in the ventral wall of the duodenum. Later, owing to the rotation of the gut, the opening is carried to the left, and then dorsalward to the position it occupies in the adult. As the liver undergoes enlargement, both it and the ventral mesogastrium of the foregut are gradually differentiated from the septum transversum, and from the undersurface of the latter, the liver projects downward into the abdominal cavity. By the growth of the liver, the ventral mesogastrium is divided into two parts, of which the anterior forms the falciform and coronary ligaments, and the posterior the lesser omentum. About the third month, the liver almost fills the abdominal cavity, and its left lobe is nearly as large as its right. From this period, the relative development of the liver is less active, more especially that of the left lobe, which actually undergoes some degeneration and becomes smaller than the right. But up to the end of fetal life, the liver remains relatively larger than in the adult. Vessels and nerves. The vessels connected with the liver are the hepatic artery, the portal vein, and the hepatic veins. The hepatic artery and portal vein, accompanied by numerous nerves, ascend to the porta between the layers of the lesser omentum. The bile duct and the lymphatic vessels descend from the porta between the layers of the same omentum. The relative positions of the three structures are as follows. The bile duct lies to the right, the hepatic artery to the left, and the portal vein behind and between the other two. They are enveloped in a loose areolar tissue, the fibrous capsule of glycine, which accompanies the vessels in their course through the portal canals in the interior of the organ. The hepatic veins convey the blood from the liver and are described on page 680. They have very little cellular investment, and what there is binds their parietes closely to the walls of the canals through which they run, so that, on section of the organ, they remain widely open and are solitary, and may be easily distinguished from the branches of the portal vein, which are more or less collapsed, and always accompanied by an artery and duct. The lymphatic vessels of the liver are described on page 711. The nerves of the liver, derived from the left vagus and sympathetic, enter at the porta and accompany the vessels and ducts to the interlobular spaces. Here, according to Korolkov, the medullated fibers are distributed almost exclusively to the coats of the blood vessels, while the non-medullated enter the lobules and ramify between the cells and even within them. Structure of the liver. The substance of the liver is composed of lobules, held together by an extremely fine areolar tissue, in which ramify the portal vein, hepatic ducts, hepatic artery, hepatic veins, lymphatics, and nerves, the whole being invested by a serous and a fibrous coat. The serous coat, tunica serosa, is derived from the peritoneum and invests the greater part of the surface of the organ. It is intimately adherent to the fibrous coat. The fibrous coat, capsula fibrosa, glissoni, areolar coat, lies beneath the serous investment and covers the entire surface of the organ. It is difficult of demonstration, except where the serous coat is deficient. At the porta it is continuous with the fibrous capsule of glycin, and on the surface of the organ with the areolar tissue separating the lobules. The lobules, lobuli hepatis, form the chief mass of the hepatic substance. They may be seen either on the surface of the organ or by making a section through the gland, as small granular bodies, about the size of a millet seed, measuring from 1 to 2.5 millimeters in diameter. In the human subject their outlines are very irregular, but in some of the lower animals, for example the pig, they are well defined, and when divided transversely have polygonal outlines. The bases of the lobules are clustered around the smallest radicles, sublobular, of the hepatic veins, to which each is connected by means of a small branch which issues from the center of the lobule, intralobular. The remaining part of the surface of each lobule is imperfectly isolated from the surrounding lobules by a thin stratum of areolar tissue, in which is contained a plexus of vessels, the interlobular plexus, and ducts. In some animals, as the pig, the lobules are completely isolated from one another by the interlobular areolar tissue. If one of the sublobular veins be laid open, the bases of the lobules may be seen through the thin wall of the vein on which they rest, arranged in a form resembling a tessellated pavement, the center of each polygonal space presenting a minute aperture, the mouth of an interlobular vein. Microscopic appearance. Each lobule consists of a mass of cells, hepatic cells, arranged in irregular radiating columns, between which are the blood channels, sinusoids. These convey the blood from the circumference to the center of the lobule, and end in the intralobular vein, which runs through its center, to open at its base into one of the sublobular veins. Between the cells are also the minute bile capillaries. Therefore, in the lobule there are all the essentials of a secreting gland, that is to say, 1. Cells, by which the secretion is formed, 2. Blood vessels, in close relation with the cells, containing the blood from which the secretion is derived, 3. Ducts, by which the secretion, when formed, is carried away. 1. The hepatic cells are polyhedral in form. They vary in size from 12 to 25 microns in diameter. They contain one or sometimes two distinct nuclei. The nucleus exhibits an intranuclear network and one or two refractile nucleoli. The cells usually contain granules, some of which are protoplasmic, while others consist of glycogen, fat, or an iron compound. In the lower vertebrates, for example frog, the cells are arranged in tubes, with the bile duct forming the lumen and blood vessels externally. According to de Lépine, evidences of this arrangement can be found in the human liver. 2. The blood vessels. The blood in the capillary plexus around the liver cells is brought to the liver principally by the portal vein, but also to a certain extent by the hepatic artery. The hepatic artery, entering the liver at the porta with the portal vein and hepatic duct, ramifies with these vessels through the portal canals. It gives off vaginal branches, which ramify in the fibrous capsule of glycin and appear to be destined chiefly for the nutrition of the coats of the vessels and ducts. It also gives off capsular branches, which reach the surface of the organ, ending in its fibrous coat in stellate plexuses. Finally, it gives off interlobular branches, which form a plexus outside each lobule to supply the walls of the interlobular veins and the accompanying bile ducts. From this plexus, lobular branches enter the lobule and end in the network of sinusoids between the cells. The portal vein also enters at the porta and runs through the portal canals, enclosed in Glisson's capsule, dividing in its course into branches, which finally break up into a plexus, the interlobular plexus, in the interlobular spaces. These branches receive the vaginal and capsular veins, corresponding to the vaginal and capsular branches of the hepatic artery. Thus it will be seen that all the blood carried to the liver by the portal vein and hepatic artery finds its way into the interlobular plexus. From this plexus, the blood is carried into the lobule by fine branches which converge from the circumference to the center of the lobule, and are connected by transverse branches. The walls of these small vessels are incomplete, so that the blood is brought into direct relationship with the liver cells. The lining endothelium consists of irregularly branched, disconnected cells, stellate cells of Cooper. Moreover, according to Herring and Simpson, minute channels penetrate the liver cells themselves, conveying the constituents of the blood into their substance. It will be seen that the blood capillaries of the liver lobule differ structurally from capillaries elsewhere. Developmentally, they are formed by the growth of the columns of liver cells into large blood spaces or sinuses, and hence they have received the name of sinusoids. Arrived at the center of the lobule, the sinusoids empty themselves into one vein, of considerable size, which runs down the center of the lobule from apex to base, and is called the intralobular vein. At the base of the lobule, this vein opens directly into the sublobular vein with which the lobule is connected. The sublobular veins unite to form larger and larger trunks, and end at last in the hepatic veins. These converge to form 3 large trunks, which open into the inferior vena cava, while that vessel is situated in its fossa on the posterior surface of the liver. 3. The bile ducts commence by little passages in the liver cells, which communicate with canaliculi, termed intercellular biliary passages, bile capillaries. These passages are merely little channels or spaces left between the contiguous surfaces of 2 cells, or in the angle where 3 or more liver cells meet, and they are always separated from the blood capillaries by at least half the width of a liver cell. The channels thus formed radiate to the circumference of the lobule and open into the interlobular bile ducts, which run in Glisson's capsule, accompanying the portal vein and hepatic artery. These join with other ducts to form 2 main trunks, which leave the liver at the transverse fissure and by their union form the hepatic duct. Structure of the ducts. Walls of the biliary ducts consist of a connective tissue coat, in which are muscle cells arranged both circularly and longitudinally, and an epithelial layer consisting of short columnar cells resting on a distinct basement membrane. Excretory apparatus of the liver. The excretory apparatus of the liver consists of 1. The hepatic duct, formed by the junction of the 2 main ducts, which pass out of the liver at the porta, 2. The gallbladder, which serves as a reservoir for the bile, 3. The cystic duct, or the duct of the gallbladder, and 4. The common bile duct, formed by the junction of the hepatic and cystic ducts. The hepatic duct, ductus hepaticus. 2 main trunks of nearly equal size issue from the liver at the porta, one from the right, the other from the left lobe. These unite to form the hepatic duct, which passes downward and to the right for about 4 centimeters between the layers of the lesser omentum, where it is joined at an acute angle by the cystic duct, and so forms the common bile duct. The hepatic duct is accompanied by the hepatic artery and the portal vein. The gallbladder, vesica phellia. The gallbladder is a conical or pear-shaped muscular membranous sac lodged in a fossa on the under surface of the right lobe of the liver, and extending from near the right extremity of the porta to the anterior border of the organ. It is from 7 to 10 centimeters in length, 2.5 centimeters in breadth at its widest part, and holds from 30 to 35 cubic centimeters. It is divided into a fundus, body, and neck. The fundus, or broad extremity, is directed downward, forward, and to the right, and projects beyond the anterior border of the liver. The body and neck are directed upward and backward to the left. The upper surface of the gallbladder is attached to the liver by connective tissue and vessels. The under surface is covered by peritoneum, which is reflected onto it from the surface of the liver. Occasionally, the whole of the organ is invested by the serous membrane, and is then connected to the liver by a kind of mesentery. Relations. The body is in relation by its upper surface with the liver, by its undersurface with the commencement of the transverse colon, and farther back usually with the upper end of the descending portion of the duodenum, but sometimes with the superior portion of the duodenum or pyloric end of the stomach. The fundus is completely invested by peritoneum. It is in relation in front with the abdominal parietes, immediately below the ninth costal cartilage, behind with the transverse colon. The neck is narrow and curves upon itself like the letter S. At its point of connection with the cystic duct, it represents a well-marked constriction. Structure. The gallbladder consists of 3 coats, serous, fibromuscular, and mucous. The external or serous coat, tunica serosa vesicae felii, is derived from the peritoneum. It completely invests the fundus, but covers the body and neck only on their under surfaces. The fibromuscular coat, tunica muscularis vesicae felii, a thin but strong layer forming the framework of the sac, consists of dense fibrous tissue, which interlaces in all directions, and is mixed with plain muscular fibers, disposed chiefly in a longitudinal direction, a few running transversely. The internal or mucous coat, tunica mucosa vesicae felii, is loosely connected with the fibrous layer. It is generally of a yellowish-brown color and is elevated into minute ruggi. Opposite the neck of the gallbladder, the mucous membrane projects inward in the form of oblique ridges or folds, forming a sort of spiral valve. The mucous membrane is continuous through the hepatic duct with the mucous membrane lining the ducts of the liver, and through the common bile duct with the mucous membrane of the duodenum. It is covered with columnar epithelium and secretes mucin. In some animals it secretes a nucleoprotein instead of mucin. The cystic duct, ductus cysticus. The cystic duct, about 4 cm long, runs backward, downward, and to the left from the neck of the gallbladder, and joins the hepatic duct to form the common bile duct. The mucous membrane lining its interior is thrown into a series of crescentic folds, from 5 to 12 in number, similar to those found in the neck of the gallbladder. They project into the duct in regular succession, and are directed obliquely around the tube, presenting much the appearance of a continuous spiral valve. When the duct is distended, the spaces between the folds are dilated, so as to give to its exterior a twisted appearance. The common bile duct, ductus choledochus. The common bile duct is formed by the junction of the cystic and hepatic ducts. It is about 7.5 cm long and of the diameter of a goose quill. It descends along the right border of the lesser omentum, behind the superior portion of the duodenum, in front of the portal vein, and to the right of the hepatic artery. It then runs in a groove near the right border of the posterior surface of the head of the pancreas. Here it is situated in front of the inferior vena cava, and is occasionally completely embedded in the pancreatic substance. At its termination it lies for a short distance along the right side of the terminal part of the pancreatic duct, and passes with it obliquely between the mucous and muscular coats. The 2 ducts unite and open by a common orifice upon the summit of the duodenal papilla, situated at the medial side of the descending portion of the duodenum, a little below its middle, and about 7 to 10 cm from the pylorus. The short tube formed by the union of the 2 ducts is dilated into an ampulla, the ampulla of Vater. Structure. The coats of the large biliary ducts are an external or fibrous, and an internal or mucous. The fibrous coat is composed of strong fibroareolar tissue with a certain amount of muscular tissue, arranged for the most part in a circular manner around the duct. The mucous coat is continuous with the lining membrane of the hepatic ducts and the gallbladder, and also with that of the duodenum, and, like the mucous membrane of these structures, its epithelium is of columnar variety. It is provided with numerous mucous glands, which are lobulated and open by minute orifices scattered irregularly in the larger ducts. End of section 24. Of Gray's Anatomy, Part 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body, Part 5, by Henry Gray. The Pancreas. The pancreas is a compound racemose gland, analogous in its structures to the salivary glands, though softer and less compactly arranged than those organs. Its secretion, the pancreatic juice, carried by the pancreatic duct to the duodenum, is an important digestive fluid. In addition, the pancreas has an important internal secretion, probably elaborated by the cells of Langerhans, which is taken up by the bloodstream and is concerned with sugar metabolism. It is long and irregularly prismatic in shape. Its right extremity, being broad, is called the head, and is connected to the main portion of the organ or body by a slight constriction, the neck, while its left extremity gradually tapers to form the tail. It is situated transversely across the posterior wall of the abdomen, at the back of the epigastric and left hypochondriac regions. Its length varies from 12.5 to 15 centimeters, and its weight from 60 to 100 grams. Relations. The head, caput pancreatis, is flattened from before backward, and is lodged within the curve of the duodenum. Its upper border is overlapped by the superior part of the duodenum, and its lower overlaps the horizontal part. Its right and left borders overlap in front and insinuate themselves behind, the descending and ascending parts of the duodenum, respectively. The angle of junction of the lower and left lateral borders forms a prolongation, termed the uncinate process. In the groove between the duodenum and the right lateral and lower borders in front are the anastomosing superior and inferior pancreatico-duodenal arteries. The common bile duct descends behind, close to the right border, to its termination in the descending part of the duodenum. Anterior surface. The greater part of the right half of this surface is in contact with the transverse colon, only areolar tissue intervening. From its upper part the neck springs, its right limit being marked by a groove for the gastroduodenal artery. The lower part of the right half, below the transverse colon, is covered by peritoneum continuous with the inferior layer of the transverse mesocolon, and is in contact with the coils of the small intestine. The superior mesenteric artery passes down in front of the left half across the uncinate process. The superior mesenteric vein runs upward on the right side of the artery, and, behind the neck, joins with the lienal vein to form the portal vein. Posterior surface. The posterior surface is in relation with the inferior vena cava, the common bile duct, the renal veins, the right crus of the diaphragm, and the aorta. The neck springs from the right upper portion of the front of the head. It is about 2.5 centimeters long, and is directed at first upward and forward, and then upward and to the left, to join the body. It is somewhat flattened from above downward and backward. Its antero-superior surface supports the pylorus. Its postero-inferior surface is in relation with the commencement of the portal vein. On the right it is grooved by the gastroduodenal artery. The body, corpus pancreatis, is somewhat prismatic in shape and has three surfaces, anterior, posterior, and inferior. The anterior surface, fasces anterior, is somewhat concave and is directed forward and upward. It is covered by the postero-inferior surface of the stomach, which rests upon it, the two organs being separated by the omental bursa. Where it joins the neck there is a well-marked prominence, the tuber omentale, which abuts against the posterior surface of the lesser omentum. The posterior surface, fasces posterior, is devoid of peritoneum, and is in contact with the aorta, the lienal vein, the left kidney and its vessels, the left suprarenal gland, the origin of the superior mesenteric artery, and the crura of the diaphragm. The inferior surface, fasces inferior, is narrow on the right but broader on the left, and is covered by the peritoneum. It lies upon the duodeno-jejunal flexure and on some coils of the jejunum. Its left extremity rests on the left colic flexure. The superior border, margo superior, is blunt and flat to the right, narrow and sharp to the left, near the tail. It commences on the right in the omental tuberosity and is in relation with the celiac artery, from which the hepatic artery courses to the right just above the gland, while the lienal artery runs toward the left in a groove along this border. The anterior border, margo anterior, separates the anterior from the inferior surface, and along this border the two layers of the transverse mesocolon diverge from one another, one passing upward over the anterior surface, the other backward over the inferior surface. The inferior border, margo inferior, separates the posterior from the inferior surface. The superior mesenteric vessels emerge under its right extremity. The tail, cauda pancreatis, is narrow, it extends to the left as far as the lower part of the gastric surface of the spleen, lying in the phrenico-lional ligament, and it is in contact with the left colic flexure. Birmingham described the body of the pancreas as projecting forward as a prominent ridge into the abdominal cavity and forming part of a shelf on which the stomach lies. The portion of the pancreas to the left of the middle line has a very considerable anterior-posterior thickness. As a result, the anterior surface is of considerable extent. It looks strongly upward and forms a large and important part of the shelf. As the pancreas extends to the left, toward the spleen, it crosses the upper part of the kidney and is so molded onto it that the top of the kidney forms an extension inward and backward of the upper surface of the pancreas and extends the bed in this direction. On the other hand, the extremity of the pancreas comes in contact with the spleen in such a way that the plane of its upper surface runs with little interruption upward and backward into the concave gastric surface of the spleen, which completes the bed behind and to the left, and, running upward, forms a partial cap for the wide end of the stomach. Footnote: Journal of Anatomy and Physiology, Part 1, Volume 31, page 102. The pancreatic duct, Ductus pancreaticus, Wersungi, duct of Wirsung, extends transversely from left to right through the substance of the pancreas. It commences by the junction of the small ducts of the lobules situated in the tail of the pancreas, and running from left to right through the body, it receives the ducts of the various lobules composing the gland. Considerably augmented in size, it reaches the neck, and turning downward, backward, and to the right, it comes into relation with the common bile duct, which lies to its right side. Leaving the head of the gland, it passes very obliquely through the mucous and muscular coats of the duodenum, and ends by an orifice common to it and the common bile duct upon the summit of the duodenal papilla, situated at the medial side of the descending portion of the duodenum, 7.5 to 10 cm below the pylorus. The pancreatic duct near the duodenum is about the size of an ordinary quill. Sometimes the pancreatic duct and the common bile duct open separately into the duodenum. Frequently there is an additional duct, which is given off from the pancreatic duct in the neck of the pancreas and opens into the duodenum about 2.5 cm above the duodenal papilla. It receives the ducts from the lower part of the head and is known as the accessory pancreatic duct, duct of Santorini. Development. The pancreas is developed in two parts, a dorsal and a ventral. The former arises as a diverticulum from the dorsal aspect of the duodenum, a short distance above the hepatic diverticulum, and growing upward and backward into the dorsal megagastrium, forms a part of the head and uncinate process and the whole of the body and tail of the pancreas. The ventral part appears in the form of a diverticulum from the primitive bile duct and forms the remainder of the head and uncinate process of the pancreas. The duct of the dorsal part, accessory pancreatic duct, therefore opens independently into the duodenum, while that of the ventral part, pancreatic duct, opens with the common bile duct. About the 6th week, the two parts of the pancreas meet and fuse, and a communication is established between their ducts. After this has occurred, the terminal part of the accessory duct, that is, the part between the duodenum and the point of meeting of the two ducts, undergoes little or no enlargement, while the pancreatic duct increases in size and forms the main duct of the gland. The opening of the accessory duct into the duodenum is sometimes obliterated, and even when it remains patent, it is probable that the whole of the pancreatic secretion is conveyed through the pancreatic duct. At first, the pancreas is directed upward and backward between the two layers of the dorsal mesogastrium, which give to it a complete peritoneal investment, and its surfaces look to the right and left. With the change in the position of the stomach, the dorsal mesogastrium is drawn downward and to the left, and the right side of the pancreas is directed backward and the left forward. The right surface becomes applied to the posterior abdominal wall, and the peritoneum which covered it undergoes absorption, and thus, in the adult, the gland appears to lie behind the peritoneal cavity. Structure. In structure, the pancreas resembles the salivary glands. It differs from them, however, in certain particulars, and is looser and softer in its texture. It is not enclosed in a distinct capsule, but is surrounded by areolar tissue, which dips into its interior and connects together the various lobules of which it is composed. Each lobule, like the lobules of the salivary glands, consists of one of the ultimate ramifications of the main duct, ending in a number of cecal pouches or alveoli, which are tubular and somewhat convoluted. The minute ducts connected with the alveoli are narrow and lined with flattened cells. The alveoli are almost completely filled with secreting cells, so that scarcely any lumen is visible. In some animals, spindle-shaped cells occupy the center of the alveolus and are known as the centroacinar cells of Langerhans. These are prolongations of the terminal ducts. The true secreting cells which line the wall of the alveolus are very characteristic. They are columnar in shape and present two zones, an outer one, clear and finely striated, next to the basement membrane, and an inner granular one next to the lumen. In hardened specimens the outer zone stains deeply with various dyes, whereas the inner zone stains slightly. During activity, the granular zone gradually diminishes in size, and when exhausted is only seen as a small area next to the lumen. During the resting stages it gradually increases until it forms nearly 3/4 of the cell. In some of the secreting cells of the pancreas is a spherical mass, staining more easily than the rest of the cell. This is termed the paranucleus, and is believed to be an extension from the nucleus. The connective tissue between the alveoli presents in certain parts collections of cells, which are termed intra-alveolar cell islets, islands of Langerhans. The cells of these stain lightly with hematoxylin or carmine, and are more or less polyhedral in shape, forming a network in which ramify many capillaries. There are two main types of cells in the islets, distinguished as A cells and B cells according to the special staining reactions of the granules they contain. The cell islets have been supposed to produce the internal secretion of the pancreas which is necessary for carbohydrate metabolism, but numerous researches have so far failed to elucidate their real function. The walls of the pancreatic duct are thin, consisting of two coats, an external fibrous and an internal mucus. The latter is smooth and furnished near its termination with a few scattered follicles. Vessels and nerves. The arteries of the pancreas are derived from the lional and the pancreatico-duodenal branches of the hepatic and superior mesenteric. Its veins open into the lional and superior mesenteric veins. Its lymphatics are described on page 711. Its nerves are filaments from the lional plexus. End of section 25.
Section 26 of Gray's Anatomy, Part 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Bianca. Anatomy of the Human Body, Part 5, by Henry Gray. Development of Urinary and Generative Organs, Part 1. The urogenital apparatus. Apparatus urogenitalis, urogenital organs. The urogenital apparatus consists of A. The urinary organs, for the secretion and discharge of the urine, and B. The genital organs, which are concerned with the process of reproduction. A. Development of the urinary and generative organs. The urinary and generative organs are developed from the intermediate cell mass, which is situated between the primitive segments and the lateral plates of mesoderm. The permanent organs of the adult are preceded by a set of structures which are purely embryonic and which, with the exception of the ducts, disappear almost entirely before the end of fetal life. These embryonic structures are, on either side, the pronephros, the mesonephros, the metanephros, and the Wolffian and Mullerian ducts. The pronephros disappears very early. The structural elements of the mesonephros mostly degenerate, but in their place is developed the genital gland, in association with which the Wolffian duct remains as the duct of the male genital gland, the Mullerian as that of the female. Some of the tubules of the metanephros form part of the permanent kidney. The pronephros and Wolffian duct. In the outer part of the intermediate cell mass, immediately under the ectoderm, in the region from the 5th cervical to the 3rd thoracic segments, a series of short evaginations from each segment grows dorsalward and extends caudalward, fusing successively from before backward to form the pronephric duct. This continues to grow caudalward until it opens into the ventral part of the cloaca. Beyond the pronephros, it is termed the Wolffian duct. The original evaginations form a series of transverse tubules, each of which communicates by means of a funnel-shaped ciliated opening with the coelomic cavity, and in the course of each duct a glomerulus also is developed. A secondary glomerulus is formed ventral to each of these, and the complete group constitutes the pronephros. The pronephros undergoes rapid atrophy and disappears. The mesonephros, Mullerian duct, and genital gland. On the medial side of the Wolffian duct, from the 6th cervical to the 3rd lumbar segments, a series of tubules, the Wolffian tubules, is developed. At a later stage in development, they increase in number by outgrowth from the original tubules. These tubules first appear as solid masses of cells, which later become hollowed in the center. One end grows toward and finally opens into the Wolffian duct. The other dilates and is invaginated by a tuft of capillary blood vessels to form a glomerulus. The tubules collectively constitute the mesonephros or Wolffian body. By the 5th or 6th week, this body forms an elongated spindle-shaped structure, termed the urogenital fold, which projects into the coelomic cavity at the side of the dorsal mesentery, reaching from the septum transversum in front to the 5th lumbar segment behind. In this fold, the reproductive glands are developed. The Wolffian bodies persist and form the permanent kidneys in fishes and amphibians, but in reptiles, birds, and mammals, they atrophy and for the most part disappear coincidentally with the development of the permanent kidneys. The atrophy begins during the 6th or 7th week and rapidly proceeds, so that by the beginning of the 5th month, only the ducts and a few of the tubules remain. In the male, the Wolffian duct persists and forms the tube of the epididymis, the ductus deferens, and the ejaculatory duct, while the seminal vesicle arises during the 3rd month as a lateral diverticulum from its hinder end. A large part of the head end of the mesonephros atrophies and disappears. Of the remainder, the anterior tubules form the efferent ducts of the testis, while the posterior tubules are represented by the ductuli aberrantis and by the paradidymis, which is sometimes found in front of the spermatic cord above the head of the epididymis. In the female, the Wolffian bodies and ducts atrophy. The remains of the Wolffian tubules are represented by the epiophoron, or organ of Rosenmüller, and the paraophoron, two small collections of rudimentary blind tubules which are situated in the mesosalpinx. The lower part of the Wolffian duct disappears, while the upper part persists as the longitudinal duct of the epiophoron, or duct of Gärtner. Footnote: Barry Hart has described the Wolffian ducts as ending at the site of the future hymen in bulbous enlargements, which he has named the Wolffian bulbs, and states that the hymen is formed by these bulbs, aided by a special involution from below of the cells lining the urogenital sinus. He further believes that the lower third of the vagina is due to the coalescence of the upper portion of the urogenital sinus and the lower ends of the Wolffian ducts, and that the epithelial lining of the vagina is derived from the Wolffian bulbs. He also regards the colliculus seminalis of the male urethra as being formed from the lower part of the Wolffian ducts. End footnote. The Mullerian ducts. Shortly after the formation of the Wolffian ducts, a second pair of ducts is developed. These are named the Mullerian ducts. Each arises on the lateral aspect of the corresponding Wolffian duct as a tubular invagination of the cells lining the coelom. The orifice of the invagination remains patent and undergoes enlargement and modification to form the abdominal ostium of the uterine tube. The ducts pass backward lateral to the Wolffian ducts, but toward the posterior end of the embryo they cross to the medial side of these ducts, and thus come to lie side by side between and behind the latter, the four ducts forming what is termed the genital cord. The Mullerian ducts and in an epithelial elevation, the Mullerian eminence, on the ventral part of the cloaca, between the orifices of the Wolffian ducts. At a later date, they open into the cloaca in this situation. In the male, the Mullerian ducts atrophy, but traces of their anterior ends are represented by the appendices testes, hydatids of Morgagni, while their terminal fused portions form the utriculus in the floor of the prostatic portion of the urethra. In the female, the Mullerian ducts persist and undergo further development. The portions, which lie in the genital cord, fuse to form the uterus and vagina. The parts in front of this cord remain separate, and each forms the corresponding uterine tube, the abdominal ostium of which is developed from the anterior extremity of the original tubular invagination from the coelom. The fusion of the Mullerian ducts begins in the 3rd month, and the septum formed by their fused medial walls disappears from below upward, and thus the cavities of the vagina and uterus are produced. About the 5th month, an annular constriction marks the position of the neck of the uterus, and after the 6th month, the walls of the uterus begin to thicken. For a time, the vagina is represented by a solid rod of epithelial cells. A ring-like outgrowth of this epithelium occurs at the lower end of the uterus and marks the future vaginal fornices. About the 5th or 6th month, the lumen of the vagina is produced by the breaking down of the central cells of the epithelium. The hymen represents the remains of the Mullerian eminence. Genital Glands. The first appearance of the genital gland is essentially the same in the two sexes, and consists in a thickening of the epithelial layer which lines the peritoneal cavity on the medial side of the urogenital fold. The thick plate of epithelium extends deeply, pushing before it the mesoderm and forming a distinct projection. This is termed the genital ridge, and from it the testis in the male and the ovary in the female are developed. At first, the mesonephros and genital ridge are suspended by a common mesentery, but as the embryo grows, the genital ridge gradually becomes pinched off from the mesonephros, with which it is at first continuous, though it still remains connected to the remnant of this body by a fold of peritoneum, the mesorchium or mesovarium. About the 7th week, the distinction of sex in the genital ridge begins to be perceptible. The ovary. The ovary, thus formed from the genital ridge, is at first a mass of cells derived from the coelomic epithelium. Later, the mass is differentiated into a central part, or medulla, covered by a surface layer, the germinal epithelium. Between the cells of the germinal epithelium, a number of larger cells, the primitive ova, are formed, and these are carried into the subjacent stroma by bud-like ingrowth, genital cords, of the germinal epithelium. The surface epithelium ultimately forms the permanent epithelial covering of this organ. It soon loses its connection with the central mass, and the tunica albuginea develops between them. The ova are chiefly derived from the cells of the central mass. These are separated from one another by the growth of connective tissue in an irregular manner. Each ovum assumes a covering of connective tissue, follicle cells, and in this way the rudiments of the ovarian follicles are formed. According to Beard, the primitive ova are early set apart during the segmentation of the ovum and migrate into the germinal ridge. Waldeyer taught that the primitive germ cells are derived from the germinal epithelium covering the genital ridge. Beard, on the other hand, maintains that in the scate they are not derived from this epithelium, but are probably formed during the later stages of cell cleavage, before there is any trace of an embryo, and a similar view was advanced by Nussbaum as to their origin in amphibia. Beard says, at the close of segmentation, many of the future germ cells lie in the segmentation cavity just beneath the site of the future embryo, and there is no doubt they subsequently wander into it. The germ cells, after they enter the resting phase, are sharply marked off from the cells of the embryo by entire absence of mitosis among them. They can be further recognized by their irregular form and amoeboid processes, and by the fact that their cytoplasm has no affinity for ordinary stains, but assumes a brownish tinge when treated by osmic acid. The path along which they travel into the embryo is a very definite one, namely, from the yolk sac upward, between the splanchnopleur and gut, in the hinder portion of the embryo. This pathway, named by Beard the germinal path, leads them directly to the position which they ought finally to take up in the germinal ridge or nidus. A considerable number apparently never reach their proper destination, since vagrant germ cells are found in all sorts of places, but more particularly on the mesentery. Some of these may possibly find their way into the germinal ridge. Some probably undergo atrophy, while others may persist and become the seat of dermoid tumors. End of section 26. Recorded by Bianca in Utrecht, the Netherlands, on February 2nd, 2010. Section 27 of Gray's Anatomy, Part 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Bianca. Anatomy of the Human Body, Part 5, by Henry Gray. Development of Urinary and Generative Organs, Part 2. The testis. The testis is developed in much the same way as the ovary. Like the ovary, in its earliest stages, it consists of a central mass of epithelium covered by a surface epithelium. In the central mass, a series of cords appear, and the periphery of the mass is converted into the tunica albuginea, thus excluding the surface epithelium from any part in the formation of the tissue of the testis. The cords of the central mass run together toward the future hilus and form a network which ultimately becomes the rete testis. From the cords, the seminiferous tubules are developed, and between them connective tissue septa extend. The seminiferous tubules become connected with outgrowths from the Wolffian body, which, as before mentioned, form the efferent ducts of the testis. Descent of the testes. The testes, at an early period of fetal life, are placed at the back part of the abdominal cavity, behind the peritoneum, and each is attached by a peritoneal fold, the mesorchium, to the mesonephros. From the front of the mesonephros, a fold of peritoneum, termed the inguinal fold, goes forward to meet and fuse with a peritoneal fold, the inguinal crest, which grows backward from the anterolateral abdominal wall. The testis thus acquires an indirect connection with the anterior abdominal wall, and at the same time a portion of the peritoneal cavity lateral to these fused folds is marked off as the future saccus vaginalis. In the inguinal crest, a peculiar structure, the gubernaculum testis, makes its appearance. This is at first a slender band, extending from that part of the skin of the groin which afterward forms the scrotum, through the inguinal canal to the body and epididymis of the testis. As development advances, the peritoneum enclosing the gubernaculum forms two folds, one above the testis and the other below it. The one above the testis is the plica vascularis and contains ultimately the internal spermatic vessels. The one below, the plica gubernatrix, contains the lower part of the gubernaculum, which has now grown into a thick cord. It ends below at the abdominal inguinal ring in a tube of peritoneum, the saccus vaginalis, which protrudes itself down the inguinal canal. By the 5th month, the lower part of the gubernaculum has become a thick cord, while the upper part has disappeared. The lower part now consists of a central core of unstriped muscle fiber, and outside this of a firm layer of striped elements, connected, behind the peritoneum, with the abdominal wall. As the scrotum develops, the main portion of the lower end of the gubernaculum is carried, with the skin to which it is attached, to the bottom of this pouch. Other bands are carried to the medial side of the thigh and to the perineum. The tube of peritoneum, constituting the saccus vaginalis, projects itself downward into the inguinal canal and emerges at the cutaneous inguinal ring, pushing before it a part of the obliquus internus and the aponeurosis of the obliquus externus, which form, respectively, the cremaster muscle and the intercrural fascia. It forms a gradually elongating pouch, which eventually reaches the bottom of the scrotum, and behind this pouch the testis is drawn by the growth of the body of the fetus, for the gubernaculum does not grow commensurately with the growth of other parts, and therefore the testis, being attached by the gubernaculum to the bottom of the scrotum, is prevented from rising as the body grows, and is drawn first into the inguinal canal and eventually into the scrotum. It seems certain also that the gubernacular cord becomes shortened as development proceeds, and this assists in causing the testis to reach the bottom of the scrotum. By the end of the 8th month, the testis has reached the scrotum, preceded by the saccus vaginalis, which communicates by its upper extremity with the peritoneal cavity. Just before birth, the upper part of the saccus vaginalis usually becomes closed, and this obliteration extends gradually downward to within a short distance of the testis. The process of peritoneum surrounding the testis is now entirely cut off from the general peritoneal cavity and constitutes the tunica vaginalis. Descent of the ovaries. In the female, there is also a gubernaculum, which effects a considerable change in the position of the ovary, though not so extensive a change as in that of the testis. The gubernaculum in the female lies in contact with the fundus of the uterus and contracts adhesions to this organ, and thus the ovary is prevented from descending below this level. The part of the gubernaculum between the ovary and the uterus becomes ultimately the proper ligament of the ovary, while the part between the uterus and the labium majus forms the round ligament of the uterus. A pouch of peritoneum, analogous to the saccus vaginalis in the male, accompanies it along the inguinal canal. It is called the canal of Nuck. In rare cases, the gubernaculum may fail to contract adhesions to the uterus, and then the ovary descends through the inguinal canal into the labium majus, and under these circumstances its position resembles that of the testis. The metanephros and the permanent kidney. The rudiments of the permanent kidneys make their appearance about the end of the 1st or the beginning of the 2nd month. Each kidney has a twofold origin, part arising from the metanephros and part as a diverticulum from the hind end of the Wolffian duct, close to where the latter opens into the cloaca. The metanephros arises in the intermediate cell mass, caudal to the mesonephros, which it resembles in structure. The diverticulum from the Wolffian duct grows dorsalward and forward along the posterior abdominal wall, where its blind extremity expands and subsequently divides into several buds, which form the rudiments of the pelvis and calyces of the kidney. By continued growth and subdivision, it gives rise to the collecting tubules of the kidney. The proximal portion of the diverticulum becomes the ureter. The secretory tubules are developed from the metanephros, which is molded over the growing end of the diverticulum from the Wolffian duct. The tubules of the metanephros, unlike those of the pronephros and mesonephros, do not open into the Wolffian duct. One end expands to form a glomerulus, while the rest of the tubule rapidly elongates to form the convoluted and straight tubules, the loops of Henle, and the connecting tubules. These last join and establish communications with the collecting tubules derived from the ultimate ramifications of the diverticulum from the Wolffian duct. The mesoderm around the tubules becomes condensed to form the connective tissue of the kidney. The ureter opens at first into the hind end of the Wolffian duct. After the 6th week, it separates from the Wolffian duct and opens independently into the part of the cloaca which ultimately becomes the bladder. The secretory tubules of the kidney become arranged into pyramidal masses or lobules, and the lobulated condition of the kidneys exists for some time after birth, while traces of it may be found even in the adult. The kidney of the ox and many other animals remains lobulated throughout life. The urinary bladder. The bladder is formed partly from the endodermal cloaca and partly from the ends of the Wolffian ducts. The allantois takes no share in its formation. After the separation of the rectum from the dorsal part of the cloaca, the ventral part becomes subdivided into three portions. 1. An anterior vesico-urethral portion, continuous with the allantois. Into this portion the Wolffian ducts open. 2. An intermediate narrow channel, the pelvic portion. And 3. A posterior phallic portion, closed externally by the urogenital membrane. The second and third parts together constitute the urogenital sinus. The vesico-urethral portion absorbs the ends of the Wolffian ducts and the associated ends of the renal diverticula, and these give rise to the trigone of the bladder and part of the prostatic urethra. The remainder of the vesico-urethral portion forms the body of the bladder and part of the prostatic urethra. Its apex is prolonged to the umbilicus as a narrow canal, which later is obliterated and becomes the medial umbilical ligament, Urachus. The prostate. The prostate originally consists of two separate portions, each of which arises as a series of diverticular buds from the epithelial lining of the urogenital sinus and vesico-urethral part of the cloaca between the 3rd and 4th months. These buds become tubular and form the glandular substance of the two lobes, which ultimately meet and fuse behind the urethra and also extend to its ventral aspect. The isthmus or middle lobe is formed as an extension of the lateral lobes between the common ejaculatory ducts and the bladder. Skene's ducts in the female urethra are regarded as the homologues of the prostatic glands. The bulbourethral glands of Cowper in the male and greater vestibular glands of Bartholin in the female also arise as diverticula from the epithelial lining of the urogenital sinus. The external organs of generation. As already stated, the cloacal membrane, composed of ectoderm and endoderm, originally reaches from the umbilicus to the tail. The mesoderm extends to the midventral line for some distance behind the umbilicus and forms the lower part of the abdominal wall. It ends below in a prominent swelling, the cloacal tubercle. Behind this tubercle, the urogenital part of the cloacal membrane separates the ingrowing sheets of mesoderm. The first rudiment of the penis, or clitoris, is a structure termed the phallus. It is derived from the phallic portion of the cloaca, which has extended on to the end and sides of the undersurface of the cloacal tubercle. The terminal part of the phallus, representing the future glands, becomes solid. The remainder, which is hollow, is converted into a longitudinal groove by the adsorption of the urogenital membrane. In the female, a deep groove forms around the phallus and separates it from the rest of the cloacal tubercle, which is now termed the genital tubercle. The sides of the genital tubercle grow backward as the genital swellings, which ultimately form the labia majora. The tubercle itself becomes the mons pubis. The labia minora arise by the continued growth of the lips of the groove on the undersurface of the phallus. The remainder of the phallus forms the clitoris. In the male, the early changes are similar, but the pelvic portion of the cloaca undergoes much greater development, pushing before it the phallic portion. The genital swellings extend around between the pelvic portion and the anus and forms a scrotal area. During the changes associated with the descent of the testis, this area is drawn out to form the scrotal sacs. The penis is developed from the phallus. As in the female, the urogenital membrane undergoes adsorption, forming a channel on the undersurface of the phallus. This channel extends only as far forward as the corona glandis. The corpora cavernosa of the penis or clitoris and of the urethra arise from the mesodermal tissue in the phallus. They are at first dense structures, but later vascular spaces appear in them, and they gradually become cavernous. The prepuce in both sexes is formed by the growth of a solid plate of ectoderm into the superficial part of the phallus. On coronal section, this plate presents the shape of a horseshoe. By the breaking down of its more centrally situated cells, the plate is split into two lamellae, and the cutaneous fold, the prepuce, is liberated and forms a hood over the glans. Adherent prepuce is not an adhesion really, but a hindered central desquamation. Very hard. The urethra. As already described, in both sexes, the phallic portion of the cloaca extends on to the under surface of the cloacal tubercle as far forward as the apex. At the apex, the walls of the phallic portion come together and fuse, the lumen is obliterated, and the solid plate, the urethral plate, is formed. The remainder of the phallic portion is for a time tubular, and then, by the absorption of the urogenital membrane, it establishes a communication with the exterior. This opening is the primitive urogenital ostium, and it extends forward to the corona glandis. In the female, this condition is largely retained. The portion of the groove on the clitoris broadens out, while the body of the clitoris enlarges, and thus the adult urethral opening is situated behind the base of the clitoris. In the male, by the greater growth of the pelvic portion of the cloaca, a longer urethra is formed, and the primitive ostium is carried forward with the phallus, but it still ends at the corona glandis. Later it closes from behind forward. Meanwhile, the urethral plate of the glans breaks down centrally to form a median groove, continuous with the primitive ostium. This groove also closes from behind forward, so that the external urethral opening is shifted forward to the end of the glans. End of section 27. Recorded by Bianca in Utrecht, the Netherlands, on March 1st, 2010.
Section 28 of Gray's Anatomy, Part 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body, Part 5, by Henry Gray. The Kidneys, Part 1. The urinary organs. The urinary organs comprise the kidneys, which secrete the urine, the ureters or ducts which convey urine to the urinary bladder, where it is for a time retained, and the urethra, through which it is discharged from the body. 1. The kidneys, renes. The kidneys are situated in the posterior part of the abdomen, one on either side of the vertebral column, behind the peritoneum, and surrounded by a mass of fat and loose areolar tissue. Their upper extremities are on a level with the upper border of the 12th thoracic vertebra, their lower extremities on a level with the 3rd lumbar. The right kidney is usually slightly lower than the left, probably on account of the vicinity of the liver. The long axis of each kidney is directed downward and lateralward, the transverse axis backward and lateralward. Each kidney is about 11.25 centimeters in length, 5 to 7.5 centimeters in breadth, and rather more than 2.5 centimeters in thickness. The left is somewhat longer and narrower than the right. The weight of the kidney in the adult male varies from 125 to 170 grams, in the adult female from 115 to 155 grams. The combined weight of the two kidneys in proportion to that of the body is about 1 to 240. The kidney has a characteristic form and presents for examination two surfaces, two borders, and an upper and lower extremity. Relations. The anterior surface, facies anterior, of each kidney is convex and looks forward and lateralward. Its relations to adjacent viscera differ so completely on the two sides that separate descriptions are necessary. Anterior surface of right kidney. A narrow portion at the upper extremity is in relation with the right suprarenal gland. A large area just below this, and involving about 3/4 of the surface, lies in the renal impression on the inferior surface of the liver, and a narrow but somewhat variable area near the medial border is in contact with the descending part of the duodenum. The lower part of the anterior surface is in contact laterally with the right colic flexure, and medially, as a rule, with the small intestine. The areas in relation with the liver and small intestine are covered by peritoneum. The suprarenal, duodenal, and colic areas are devoid of peritoneum. Anterior surface of the left kidney. A small area along the upper part of the medial border is in relation with the left suprarenal gland, and close to the lateral border is a long strip in contact with the renal impression on the spleen. A somewhat quadrilateral field about the middle of the anterior surface marks the site of contact with the body of the pancreas, on the deep surface of which are the lyinal vessels. Above this is a small triangular portion between the suprarenal and splenic areas, in contact with the posterior-inferior surface of the stomach. Below the pancreatic area, the lateral part is in relation with the left colic flexure, the medial with the small intestine. The areas in contact with the stomach and spleen are covered by the peritoneum of the omental bursa, while that in relation to the small intestine is covered by the peritoneum of the general cavity. Behind the latter are some branches of the left colic vessels. The suprarenal, pancreatic, and colic areas are devoid of peritoneum. The posterior surface, facies posterior. The posterior surface of each kidney is directed backward and medialward. It is embedded in an areolar and fatty tissue and entirely devoid of peritoneal covering. It lies upon the diaphragm, the medial and lateral lumbocostal arches, the psoas major, the quadratus lumborum, and the tendon of the transversus abdominis, the subcostal, and one or two of the upper lumbar arteries, and the last thoracic, ilio-hypogastric, and ilio-inguinal nerves. The right kidney rests upon the 12th rib, the left usually on the 11th and 12th. The diaphragm separates the kidney from the pleura, which dips down to form the phrenico-costal sinus, but frequently the muscular fibers of the diaphragm are defective or absent over a triangular area immediately above the lateral lumbocostal arch, and when this is the case, the perinephric areolar tissue is in contact with the diaphragmatic pleura. Borders. The lateral border, margo lateralis, external border, is convex and is directed toward the posterolateral wall of the abdomen. On the left side, it is in contact at its upper part with the spleen. The medial border, margo medialis, internal border, is concave in the center and convex toward either extremity. It is directed forward and a little downward. Its central part presents a deep longitudinal fissure, bounded by prominent overhanging anterior and posterior lips. This fissure is named the hilum, and transmits the vessels, nerves, and ureter. Above the hilum, the medial border is in relation with the suprarenal gland, below the hilum with the ureter. Extremities. The superior extremity, extremitus superior, is thick and rounded, and is nearer the median line than the lower. It is surmounted by the suprarenal gland, which covers also a small portion of the anterior surface. The inferior extremity, extremitus inferior, is smaller and thinner than the superior, and farther from the median line. It extends to within 5 centimeters of the iliac crest. The relative position of the main structures in the hilum is as follows. The vein is in front, the artery in the middle, and the ureter behind and directed downward. Frequently, however, branches of both artery and vein are placed behind the ureter. Fixation of the kidney. The kidney and its vessels are embedded in a mass of fatty tissue termed the adipose capsule, which is thickest at the margins of the kidney and is prolonged through the hilum into the renal sinus. The kidney and the adipose capsule are enclosed in a sheath of fibrous tissue continuous with the subperitoneal fascia, and named the renal fascia. At the lateral border of the kidney, the renal fascia splits into an anterior and a posterior layer. The anterior layer is carried medialward in front of the kidney and its vessels, and is continuous over the aorta with the corresponding layer of the opposite side. The posterior layer extends medialward behind the kidney and blends with the fascia on the quadratus lumborum and psoas major, and through this fascia is attached to the vertebral column. Above the suprarenal gland, the 2 layers of the renal fascia fuse and unite with the fascia of the diaphragm. Below they remain separate and are gradually lost in the subperitoneal fascia of the iliac fossa. The renal fascia is connected to the fibrous tunic of the kidney by numerous tuberculi, which traverse the adipose capsule and are strongest near the lower end of the organ. Behind the fascia renalis is a considerable quantity of fat, which constitutes the paranephric body. The kidney is held in position partly through the attachment of the renal fascia and partly by the apposition of the neighboring viscera. General structure of the kidney. The kidney is invested by a fibrous tunic, which forms a firm, smooth covering to the organ. The tunic can easily be stripped off, but in doing so, numerous fine processes of connective tissue and small blood vessels are torn through. Beneath this coat, a thin, wide mesh network of unstriped muscular fiber forms an incomplete covering to the organ. When the capsule is stripped off, the surface of the kidney is found to be smooth and even and of a deep red color. In infants, fissures extending for some depth may be seen on the surface of the organ, a remnant of the lobular construction of the gland. The kidney is dense in texture, but is easily lacerable by mechanical force. If a vertical section of the kidney be made from its convex to its concave border, it will be seen that the hilum expands into a central cavity, the renal sinus. This contains the upper part of the renal pelvis and the calyces, surrounded by some fat in which are embedded the branches of the renal vessels and nerves. The renal sinus is lined by a prolongation of the fibrous tunic, which is continued around the lips of the hilum. The renal calyces, from 7 to 13 in number, are cup-shaped tubes, each of which embraces one or more of the renal papillae. They unite to form 2 or 3 short tubes, and these in turn join to form a funnel-shaped sac, the renal pelvis. The renal pelvis, wide above and narrow below where it joins the ureter, is partly outside the renal sinus. The renal calyces and pelvis form the upper expanded end of the excretory duct of the kidney. The kidney is composed of an internal medullary and an external cortical substance. The medullary substance, substantia medullaris, consists of a series of red-colored striated conical masses, termed the renal pyramids, the bases of which are directed toward the circumference of the kidney, while their apices converge toward the renal sinus, where they form prominent papillae projecting into the interior of the calyces. The cortical substance, substantia corticalis, is reddish-brown in color and soft and granular in consistence. It lies immediately beneath the fibrous tunic, arches over the bases of the pyramids, and dips in between adjacent pyramids toward the renal sinus. The parts dipping in between the pyramids are named the renal columns, Bertini, while the portions which connect the renal columns to each other and intervene between the bases of the pyramids and the fibrous tunic are called the cortical arches. If the cortex be examined with a lens, it will be seen to consist of a series of lighter-colored conical areas, termed the radiate part, and a darker-colored intervening substance, which from the complexity of its structure is named the convoluted part. The rays gradually taper toward the circumference of the kidney, and consist of a series of outward prolongations from the base of each renal pyramid. End of section 28.